RBCs or erythrocytes are the predominant cells of blood. Now, their major role is to carry oxygen from lungs and transport it to different tissues. RBCs lack mitochondria, they do not have any organelle, and they don't have any nucleus. So that means they cannot use oxygen and they cannot regenerate themselves. Hello, my name is Dr. Kamya Tikku, and in today's video, we are going to talk about protein of RBCs, the major component of RBCs, which is called hemoglobin. Hemoglobin is also written as HB. Hemoglobin is divided into two portions, heme and globin. Heme is the pigment part, which gives it the red color, and globin is the protein part. Now, hemoglobin is a type of globular protein. That means it is circular in shape, present in RBCs, which transports oxygen in our body through blood. The heme portion has iron, which is usually present in Fe2 plus form, and porphyrin. Porphyrin is a ring which is made up of 4 pyrrole rings. That's why we call it tetrapyrrole. It's a structure linked with Fe2 plus iron. Whereas globin is a tetramer made up of 2 alpha and 2 beta chains. Each alpha chain is made up of 141 amino acids, which makes the total of 282 amino acids. And each beta chain is made up of 146 amino acids, which makes a total of 292 amino acids. In total, both alpha 2 and beta 2 chains make up 574 amino acids. Now, this respiratory pigment, as I already mentioned, helps in transporting oxygen as oxyhemoglobin from lungs to different parts of the body. When we look through the structure of hemoglobin, it has 4 subunits, alpha chain 1, alpha chain 2, beta chain 1 and beta chain 2. And within these are present heme groups. These are the heme groups, 4 heme groups. Each heme group has a ring structure like this. This is a 5-membered heterocyclic ring, which is bound through methylene bridges. And such 4 rings are present. Oxygen binds reversibly to the ferrous iron atom in each heme group, and this process is called oxygenation. And the interaction between the subunits in the hemoglobin is called cooperativity. So when hemoglobin molecule has 2 states, T state, that is called tense state, and R state, that is called relaxed state. T state is a deoxyhemoglobin state. When first oxygen molecule binds to the heme unit, the deoxyhemoglobin T state causes conformational change, leading to an increase in the affinity. And thus the second molecule binds more rapidly, and this type of binding is called allosteric binding. In this binding at one site affects the binding of the remaining binding sites. So this is the T state, the deoxyhemoglobin state. You can see here the active site, which is meant for oxygen binding, is not exposed yet. When oxygen molecule comes, first oxygen molecule binds. Binding of first oxygen molecule leads to change in the state of hemoglobin from tense state to the R state, where the active site is exposed and ready for oxygen molecule to bind for rest of the subunits also. And the binding of oxygen by hemoglobin is cooperative. With the hemoglobin binding to successive oxygens, the oxygen affinity of the subunit increases. The main function of hemoglobin is to carry and transport oxygen to various tissues, and binding of oxygen to hemoglobin in this cooperative binding helps in release of oxygen to different tissues, and also helps in transition between low oxygen affinity T state and high oxygen affinity R state. That's all for the video. Thanks for watching.